This is a recorded lecture for Chapter 4, which is the dynamics of microbial growth. In this chapter, we're going to look at how organisms grow and how the process by which they grow, the principles that they use. Now, historically, the German physicist Robert Koch, in 1843, 1911, and 1910, he studied disease-causing bacteria and won the Nobel Prize in 1905. He developed methods of cultivating bacteria. He worked on methods of solid media to allow single bacteria to grow and form colonies. He tried potatoes, but the nutrients limiting for many bacteria were kind of an inhibitory for his success. And there's a solidifying liquid nutrient media with gelatin helped. Limitations were melting temperature and digestibility of the organisms. And so in 1882, Fanny Hess, the wife associate, suggested agar that she had used to harden jelly. Now, prokaryotes can be found growing in severe conditions: ocean depths, volcanic vents, polar regions. They all harbor prokaryotic species. And many scientists believe that if life exists on other planets, it may resemble these microbes. So individual species have limited set of conditions and also require appropriate nutrients. Now, it's important to grow microbes in a culture that have medical significance and that have nutritional industrial uses.
Started.
Now, prokaryotes cells divide by binary fusion. We've talked about that before, where one cell divides into 2 into 4, 4 into 8, 8 into 16, and so forth. And so it causes what's called exponential growth. So the population literally doubles during each division. And so you see here, one organism becomes 2, and then on down the line. The generation time is the time it takes for the population to double. That's to go from here, basically, to here. And it varies among different species, and it also varies by each species, depending on the environmental conditions, the nutrients that are available, and that kind of thing. Now, exponential growth has important consequences. 10 cells of foodborne pathogen in a potato salad at a picnic can cause 40,000 cells in 4 hours. So that's pretty, when you think about how astronomically large that can become, the inoculum, the initial ingestion, if you will, of the disease can be pretty important. Now, growth can be calculated. Let's go through this long spiel of how you do that. I don't care if you know all that. Basically, what you have to do is basically say n to the t equals n to the 0 with 2 to the nth, which is really the number of populations at the time, and nt. No is the initial number of cells, and n equals the number of generations at that point. So the example is a pathogen in a potato salad at a picnic in the sun assumes 10 cells with a 20-minute generation time. So n equals 10 cells in the original population. The n itself includes 12. There's 3 divisions per hour for 4 hours. And so NO times 2N, which equals 10 times 2 to the 12, equals 10 times 4,096, which means that would be 40,960 cells in a pathogen with a pathogen in 4 hours. Pretty extraordinary when you think about it. The power of exponential growth, the rapid generation time with optimal conditions can yield huge populations pretty quickly, and this gives you an idea, this table gives you an idea. Remember that generation time depends on species, growth conditions, environmental factors, and such. Now, microorganisms historically have been studied in the laboratory, but dynamic, complex conditions in nature have a profound effect on microbial growth and behavior. The cells sense changes, they adjust to surroundings, they synthesize compounds useful for growth, and most live in polysaccharide-infused communities termed as biofilms. So it causes the slipperiness of rocks and stream beds, the slimy gunk in the sink drains, the scum of the toilet bowls, dental plaque. All of that is part of this biofilm mechanism we talk about. Now, the formation of biofilm is pretty simple. You have a group of different kinds of bacteria together. They start to create these multiply and produce an extracellular polymeric substance, if you will. The other bacteria can attach to this and get included. Finally, they have these biofilms, and then when some cells attach and move other surfaces to create additional biofilms, they are released, and to release a large amount of these different kinds of bacteria that have not been picked up by the immune system. Now, biofilms have characteristic architecture, channels through which nutrients and waste pass. The cells communicate by synthesizing these chemical signals. And biofilms have pretty important implications. For instance, dental plaque leads to tooth decay and gum disease. Most infections, like ear infections and cystic fibrosis. There's industrial concern because there's accumulation of pipes and drains. Biofilm structure shields microbes growing within and maybe hundreds of times more resistant to disinfectants. So biofilms can also be helpful. They work in bioremediation, breaking down of wastewater, and in water treatment plants. Now, prokaryotes regularly grow in close association, so there are many different species. Interactions can be cooperative. In other words, they can foster growth of species otherwise unable to survive. The strict anaerobes can grow in the mouth, and others consume the oxygen. The metabolic waste of one can serve as a nutrient for another, so they can have a very symbiotic relationship. Now, interactions are often competitive. Some synthesize toxic compounds to inhibit competitors, and some gram-negative bacteria use the needle-like approach, if you will. A needle-like structure called a type 6 secretion system to inject toxic compounds directly into competing bacteria. So they basically poison their competitors. Now, pure cultures, obtaining a pure culture. So we had to grow a pure culture so we could investigate what a bacteria is or what it does. And so pure cultures have to be of one population. So pure culture is defined as population cells that are derived from a single cell. It allows the study of a single species. Pure cultures are obtained using what's called aseptic technique. An aseptic technique is a way that we eliminate contaminate ourselves, people around us, anybody else in the lab, one culture to the other, and so forth. And it minimizes potential contamination. The cells are grown in a culture medium, contains nutrients dissolved in water, and that can be in both a broth, which is in liquid form, or a solid gel, which is called an agar. Now, you need culture medium, container, aseptic conditions, and a method to separate the individual cells. And with correct conditions, the single cells will then multiply. They form a visible colony. About 1 million cells are easily visible, and the agar is used to solidify, not destroyed by high temperatures, and can be sterilized. It liquefies above 95 degrees centigrade, solidifies below 45 degrees centigrade, and few microbes can degrade it. So the growth of a petri dish allows the air to enter, excludes contaminants, and the culture medium is a petri dish called a plate. So let's talk about streak plate method, and it's the simplest, most commonly used method for isolating organisms. It spreads out cells to separate them and to create a condition where only single cells can create colonies that can then be picked up. So we have an inoculation loop here. It has a loop at the end. We heat it up so it's bright red, and then we let it cool. We stick it in the broth, or we stick it into the specimen, the culture. We have another plate with a single colony, and then we put that into one area of the petri dish. Put that in one area. Then we heat it up again, and we streak away from that. And we heat it up again, we streak away from that. So we do that again and again and again until the last streak we create is really the cells spread out as far as they can be spread out. Now, we also try to maintain stock cultures, and a streak method yields pure cultures. Now, you can see that method here. There's the first streak, second streak, third streak, and fourth streak. You can see the fourth streak has a much thinner growth. And you see this here. See the much thinner growth. So single colonies, as a matter of fact, so you can use those for developing your pure culture. And the streak plate method helps to create pure cultures. You can maintain stock cultures. They're often stored in refrigerators and agar slants. And the cells can be frozen at -7 degrees centigrade for long-term storage, cryo methodologies. It's mixed with glycerol to prevent ice crystal formation, and it can also be freeze-dried, strangely enough. Now, prokaryotes grow in agar plates or in tubes or flasks or both. These are closed systems, so the nutrients are not renewed, the wastes are not removed. And we also have batch cultures, and they yield a characteristic growth culture, so they're a little different, these mechanisms. Here's a regular growth curve, and what you're going to see is that when the organism is first introduced into its environment, it's not really doing anything. It's kind of adapting, if you will, to what's going on. And then it starts to identify the nutrients, and it grows an exponential or log phase, it's called. And it grows very quickly. At the stationary phase, what happens is it's used up a lot of its nutrients, and the death rate is the same rate as the birth rate. So it's stationary. There's many new organisms being born, or if you will, developed with growth, than there are the ones that are dying. And at a certain point, when the nutrients are all gone, the area is more toxic, there's a death phase. And the death phase is here at the end, and it's a prolonged phase of prolonged declination. So there's lag phase, exponential phase, stationary phase, and death phase. The phase of prolonged decline is really not part of the 4 phases we looked in the growth phase. Normally, the decline, the phase of prolonged decline, is really the additional 5th phase. So you'll see 4 phases and 5-phase systems. So lag phase, number of cells does not increase. You can synthesize the enzymes required for growth in a new environment. And the delay kind of depends on the conditions of where they're put. The exponential or log phase, the cells divide at a constant rate. Generation time is measured. Most sensitive to antibiotics at this point in time because the bacteria are vegetative and they're multiplying through mitosis. And the production of primary metabolites, importantly, to be important commercially. The secondary metabolites production occurs as nutrients are depleted and waste accumulates. Stationary phase is the nutrients levels are too low to sustain growth. The total numbers remain constant. Some die. Released contents, others grow, but the death rate and growth rate is the same. The death phase is really where the cells are not growing at this point in time. The cells are dying at a constant rate. It can be exponential, but usually it's much slower than the cell growth we saw before. In the phase of prolonged decline, some fraction may survive. They can adapt to tolerate the worst conditions, but probably not. Now, colony growth, colonies, and liquid culture share similarities, and there are important differences based on location. The position of a single cell determines the environment. So the edge of a colony has oxygen and nutrients, and the center of the colony is depleted as oxygen and nutrients. So accumulation of potentially toxic waste, including acids, occur at the center of a colony. A colony may range from exponential growth at edges to death phase in the center. So you may see the growth phases in a single colony based on location. An open system is required to maintain continuous growth, and it's termed a continuous culture. Nutrients are continuously added, wastes are continuously removed, and chemostats can maintain continuous growth. Continuous drips of fresh medium in the culture in the chamber. The equivalent volume is removed. It contains cells and waste and spent medium. And the nutrient content and speed of addition can be controlled so we can achieve a constant growth rate and cell density. And they produce relatively uniform populations for study. So this constant growth system works a little differently. Now, prokaryotes inhabit nearly all environments on the Earth. Some live in comfortable habitats favored by humans, and some live in harsh environments. They're termed extremophiles, and most are archaea. The major conditions that influence their growth is temperature, atmospheric pressure, the acidity or alkalinity, pH, or the water availability in that given environment. This is a list of factors that influence microbial growth. I'll not go down this list, but you can take a look at them in the table in your book. Now, temperature requirements. Each species has well-defined temperature ranges, and we identify those species, we name them based on where their optimum growth usually occurs. So here's the optimum growth is always the top of the cycle, so to speak. So it's usually close the upper range. We have psychrophiles, which tend to grow at -5 to 15 degrees centigrade. We see those in Arctic and Antarctic regions. Psychrotrophs, which grow at 20 to 30 degrees centigrade. They're important in food spoilage. Mesophiles, 25 to 45 degrees centigrade, and these are pathogens because they occur at the same temperature as our body temperature, which is 36 degrees centigrade. We have thermophiles, which grow at 45 to 70 degrees centigrade. They're common in hot springs. And then we have hyperthermophiles, which can grow actively from 70 to 110 degrees centigrade environments. They usually are members of the archaea. They're found in the hypothermal vents around geysers and such. Now, proteins of thermophiles are resist denaturing. The thermal stability comes from amino acid sequence, number of position of bonds, which determine the structure. Temperature and food preservation, refrigeration at 40 degrees centigrade slows spoilage by limiting growth of otherwise fast-growing mesophiles. Psychrophiles, psychotrophs can still grow, but they grow slowly. Freezing preserves food, but it's not effective at killing microbes. If we look at temperature and disease, the temperature of different parts of the human body differs. Some microbes cause disease in certain parts of the body. For example, Hansen's disease, leprosy, occurs in the coolest regions, the ears, the hands, the feet, the fingers, due to the preference of Mycobacterium leprae. Now, oxygen requirements, a shade tube. We boil the nutrient agar to drive off the oxygen, and we cool it to just above solidifying temperature, and we inoculate. And then we gently swirl it. And the growth demonstrates the organism's O2 requirements. It can be an obligate anaerobe, so it only grows at the top of the tube, the top of the fluid. It can be a facultative anaerobe, which means it likes to grow anywhere in the tube, even at the at the surface. You'd be an obligate anaerobe as a facultative anaerobe. An obligated anaerobe only grows in the distal end. It can't grow around oxygen. The microaerophile only grows, doesn't grow at the edge of the tube, the top of the tube, just below that, if you will. And then we have aerotolerant aerotube, which you can just pretty much grow anywhere. And you can see the differences in the growth characteristics, the use of oxygen, and typical mechanisms to prevent or protect against reactive oxygen species. Now, reactive oxygen species are using O2 in aerobic respiration, which produce harmful reactive oxygen species, ROS by byproducts. We include superoxide or O2 and hydrogen peroxide. And they're damaging the cell components, so they react to the oxygen. The cells must have mechanisms to protect themselves, and so those are like obligate anaerobes. They don't really have to worry about that because they're located in places where the oxygen isn't. Almost all organisms growing in presence of oxygen produce an enzyme, superoxide imutase, which inactivates superoxide by converting oxygen to H2O2. So almost all produce catalase, which converts H2O2 to oxygen and water. The exception is aerotolerant anaerobes, which makes for a useful test, if you will, for some of our mechanisms. Now, bacteria survive, as far as pH, bacteria survive a range of pH which is often to them. Most maintain a constant internal pH, typically they're neutral, and they pump out the protons if it's an acidic environment, and they bring in the protons if it's an alkaline environment. Most microbes are neutrophiles, which means they live at a pH range of 5 to 8, often near 7, which is neutral, and food can be preserved by increasing the acidity. H. pylori grows in the stomach, produces urease to split urea into CO2 and ammonia to decrease acidity of the surroundings. Acidophiles grow optimally at a pH of 5.5 or very acidic. Picrophilus ashiami has an optimum pH of less than 1, believe it or not. Isn't that interesting? Less than 1, almost 0, which means it grows in the highest acid environment we have. Alkaphiles grow optimally at pH above 8.5, or the more alkaline range in the pH scale. Now we look at water availability. All microorganisms require water for growth. Dissolved salts and sugars make water unavailable to the cell. If the solute concentration is higher outside the cell, the water diffuses out, called osmosis. Salt, sugar is used to preserve food. Now, what you're going to see here is a definition of how that all works. So, osmosis is the process that we talked about before of water moving through a selective membrane, permeable selective membrane, that allows the water to go through it from an area of low concentration to an area of high concentration. So it always moves from low concentration to high concentration because it's always trying to keep the two concentrations to be equal. So since it's only the water that can move, the water has to move from a low concentration area to a high concentration area outside. The environment around this would be hypertonic. We believe that hypertonic. It has more concentration inside the cell. What happens then is fluid then leaves the cell and moves outward. And what you can see is that the fluid leaves the cell. It becomes more concentrated inside, but it sort of causes the cell to go through desiccation. It may dilute the outside, but the cell usually dies. So some microbes can withstand or even require high salt. Halotolerant can withstand up to 10%, for instance, staphylococci. Halophiles require high salt concentrations like marine bacteria, and we have extreme halophiles which can require greater than 9% salt content, like the Dead Sea in Israel and Utah's salt flats, the Salton Sea in California. Now, prokaryotes have remarkable metabolic diversity. So the nutritional factors that influence microbial growth, they require nutrients to synthesize cell components. So lipid membranes, cell walls, proteins, nucleic acids. And these are made from subunits like phospholipids, sugar, amino acids, and nucleotides. And the major elements are carbon, oxygen, hydrogen, and nitrogen, sulfur, phosphorus, potassium, magnesium, calcium, and iron. And if you think about it, these are created either in the content, the substance that we create, these macromolecules, are created in our rough endoplasmic reticulum or the smooth endoplasmic reticulum. Now, the required elements, major elements to make up cell components, you have to have a carbon source, which distinguishes between different groups. Heterotrophs use organic carbon, and autotrophs use inorganic carbon, like carbon dioxide, if you will. So remember, carbon dioxide is inorganic. Even though it has carbon, it's inorganic, as is carbon monoxide. And there's a process called carbon fixation in that process. Now, nitrogen is acquired from amino acids and nucleic acids, and many use ammonia. Some convert nitrite to ammonia, and there's nitrogen fixation is important. So iron and phosphorus are often the limiting factors, And trace elements usually are available like cobalt, zinc, copper, molybdenum, and manganese. And so we see the different requirements for their growth. Growth factors. Some microbes cannot synthesize certain molecules, like amino acids or vitamins or purines or pyrimidines. They only grow when these growth factors are available, and it reflects the biosynthetic capabilities. For instance, E. coli synthesizes all cellular components from glucose and has a wide metabolic capability to grow almost anywhere, as opposed to Neisseria, which is unable to synthesize many required numerous growth factors for itself. When organisms are fastidious, the term fastidious, they have complicated nutritional requirements like the Neisseria, for instance. The E. coli would be non-fastidious. They also have energy sources. They can be sunlight or chemical compounds. Phototrophs obtain energy from sunlight. They're photosynthesis, plants, algae, photosynthetic bacteria. Chemotrophs extract energy from the chemicals, like mammalian cells, fungi, many types of prokaryotes. They extract sugars, amino acids, fatty acids are common sources. And some prokaryotes use inorganic chemicals such as hydrogen sulfide or even hydrogen gas. So those are different mechanisms for obtaining that energy we talked about. There's nutritional diversity, which is based on the phototrophs, which use energy from sunlight and carbon from CO2. We have the photoheterotrophs, which get energy from sunlight but carbon from organic compounds. We have the chemolithoautotrophs, which are also termed chemoautotrophs or chemolithotrophs. They get energy from inorganic compounds and carbon from CO2. And the chemoorganoheterotrophs, also termed chemoheterotrophs or chemoorganotrophs, they get energy and carbon from the organic compounds. This talks about where they get their energy and where they get their carbon. They need both to survive and to grow. We cultivate microorganisms in the laboratory. We have general categories of culture media. We have complex media, which contains a variety of ingredients. Its exact composition is highly variable, and often a digest of proteins. We have chemically defined media, which is composed of exact amounts of pure chemicals. It's used for specific research requirements, and it's usually buffered. And you can see here there are different types of media. We have blood agar, chocolate agar, glucose salts, McConkey's agar, nutrient agar, and Thayer Martins are different types. And you look at the book to see what the definitions of those are. So cultivating prokaryotes in the laboratory, there's hundreds of types of media available. Regardless, some medically important microbes and most environmental ones have not yet been grown in the laboratory. They're not able to do that. There's the blood agar, chocolate agar, glucose salts, McConkey's agar, nutrient agar, and Thayer Martin we talked about before. Now, there's special types of culture media which are used for isolating and identifying specific species. Selective media inhibits the growth of certain species, so it only allows some to grow. Differential media contains substances that the microbes change in a variety of ways. For instance, here we have a blood agar plate. We have no hemolysis, so it doesn't break down the red blood cells. Here we have alpha hemolysis. We break them down, but not as completely. And then we have beta hemolysis, where you actually see through the plate because it creates a clearing of the cells. Difference, and that tells us what different kinds of bacteria we have in that group. They can be aerobic or mostly obligate aerobes, and facultative anaerobes can be incubated in air about 20% oxygen. Broth culture shaken to provide a maximum aeration. Many medically important bacteria like Neisseria and Monophilus, they grow best with increased CO2. And so we try to increase the CO2. We do that through chemical mechanisms where we drop it in the jar and it uses up all the O2, or increase more CO2, or we have what's called a candle jar. We put the plates in the jar, we put a candle in it, we light the candle eats up all the oxygen, creates CO2, and then we create an environment for Neisseria. And that also works for Neisseria gonorrhea. Some are carbonophiles, which means they require increased CO2. One method is to incubate in this candle jar we talked about. Microaerophilic organisms require lower O2 concentrations and are achieved by the candle jar. They can incubate in a gas-tight container with chemical packet, or chemical reactions reduce the O2 oxygen to less than 5 to 15%. The anaerobic are obligate anaerobes, which are sensitive to oxygen. In fact, oxygen will kill them. Anaerobic containers are useful if microbe can tolerate brief O2 exposures, but it can also use semi-solid culture in the medium containing reducing agents, sodium thioglycolate, for instance, which reduces oxygen to water. The anaerobic chamber provides a more stringent approach as far as the growth, so it doesn't kill off the bacteria when they come in contact with oxygen. We also have enrichment cultures. Enrichment cultures are used to isolate the organisms that constitute a small fraction of the mixed population. They provide conditions for the growth of particular species, for instance, certain carbon source. So you see here, we take the enrichment culture, we incubate it, and then we inoculate that to a plate and see what we're looking at, what kind of bacteria we're looking at. We also can use direct cell counts. That's where we take total numbers of living plus the dead in a direct microscopic count. We put them in what's called a hemocytometer, and we count them. We look at certain square, and we count how many organisms we see, and then we multiply that times a certain factor, and it tells us what the factor is per cubic millimeter. We also can filter them. We put a sample liquid, the bacterial cell, and the electronic detector will tell us how many organisms it counts. That's another mechanism we do. We can do viable cell counts. The cells are capable of multiplying, so we can use selective differential media for a particular species, for instance, plate counts, where a single cell gives rise to colony, and then we plate out dilution series of 30 to 300 colonies as ideal. So we have 1 mil, we add it here, and we create this 5,000 cells per mil, 3,000 cells per mil. We then take 1 mil and put it in here in a 1 to 10 dilution, and that's 5,000 cells per mil. We put it here, it's 500 per mil, here 50 per mil, and here 5 per mil. And it will show you that it helps to show you the colony counts based on that separation. The plate counts are colony forming units with CFUs, and we take a culture diluted as needed. We put that culture on the plate, we spread it out, and then we do a colony count. We can do that through factors of multiplication in the solution we created. We also have membrane filtration, which concentrates microbes by filtration. We have a filter we have here. That filter then filters out certain bacteria. The filter has a microscopic cross-hatching on it, and we can count the bacteria or the organisms that are found in a certain group of those cross-hatchings. The most probable number, MPN, is an estimation. And it estimates the cell concentration using a dilution series. So the set of tubes are incubated, results are recorded, and compared to the table to get statistical determination. So basically it tells us maybe it might be the cloudiness of a cell, it might be something else growing, but we're predicting what the content of the cell concentration is based on that growth. Measuring the biomass, turbidity is proportional to concentration of cells, so basically as the cells grow in the broth, it becomes cloudier or more turbid, and then we look at light through a spectrophotometer, which tells us how much light is getting through, and we do a calculation which will give us an estimation of the cell count in that solution. And then we can measure using total weight can be measured. It's tedious and pretty time consuming, and we don't do it too much anymore except in extreme laboratory experiments. Typically only used for filamentous organisms that do not really separate into individual cells for valid plate counts. The cells of the liquid cell culture are centrifuged. The pellet is weighed, if you will, at the bottom. The dry weight can be determined by heating the pellet in the oven. We can check cell products, for instance, the pH created by the cell, the Durham tubes, which are inverted tubes to direct gas, the CO2 production, AT produces an enzyme, luciferase to produce light. All of these are mechanisms that we use to detect the cell quantity. So what we looked at here is we looked at the dynamics of microbial growth. Thank you very much.